Hello, my name is Michelle Monje. I'm a neuroscientist and neuro-oncologist at Stanford University. I remember that on November 9th, 1989, when I was 13 years old, I was at ice skating practice. And one of my coaches came back to the ice after being on break with the very exciting news that the Berlin Wall had fallen. I have wanted to be a doctor since I was about 3 years old, and this wish did come true. And even more than that, because as a child, I had not yet imagined the sheer joy of scientific discovery, of seeking new knowledge about what is true and what is useful in nature. My research breaks the wall between brain cancer and neuroscience. Brain cancers, such as glioblastoma and diffuse intrinsic pontine glioma, are seemingly intractable malignancies. They really demand a new fundamental understanding so that we can develop better therapies. By approaching these diseases from the perspective of basic neuroscience, we've discovered new fundamental interactions that drive these cancers and hope that these new insights will result in better therapies for these terrible diseases. The essential finding of my research is that brain cancers like glioblastoma and diffuse intrinsic pontine glioma interact with normal brain cells called neurons, and that these interactions are fundamental to the progression and growth of these cancers. We find that neurons, in experience and activity-dependent manners, drives the growth of high-grade glioma cells, and that this happens both through secreted growth factors, but also really importantly, through connections called synapses. Through synapses, these contact points between cells, signals can flow, including electrical signals. And we find that neurons form functional synapses with the cancer cells. And in this way, the cancer cells integrate into neural circuits. As a result, there are electrical currents that flow through the cancers. This video shows you that this particular kind of brain cancer is an electrically active tissue. And what we find is this electrical signaling is really central to the tumor's ability to grow and invade. And we hope that by understanding this, we will be able to develop new therapies that result in better outcomes for patients who suffer from these terrible cancers. Understanding how brain activity can drive brain cancer progression opens up an entirely new direction of potential therapeutic strategies to treat these diseases. We've already brought one such strategy to clinical trial for children with glial malignancies. And it's my hope that this approach will prove complementary to traditional cancer therapies and newer immune-based therapies to affect better outcomes for children and adults suffering from these terrible brain cancers. There are so many unanswered questions. How do the cancer cells begin to integrate into the brain? Is this integration and circuit assembly between normal neurons and malignant cells similar to the way that circuits assemble during brain development? And how is it different? Does this circuit integration evolve over the course of the disease? Are normal neural circuits remodeled? How does this affect patient cognition and other forms of brain function? Most importantly, how can we target this therapeutically without disrupting normal brain function? How can we disintegrate the cancer that is integrating into the brain while leaving the normal brain intact? All of these questions remain to be answered, and they're pressing so that we can bring new therapies to people who need them. I have 4 children who have varying levels of understanding of what I do based on their age. But I will say that all of them keep me quite humble and grounded. My 10-year-old son routinely asks me when I come home from the hospital or from the lab, "Mom, have you cured brain cancer yet?" To which I always have to reply, of course, "No, honey, not yet." And then he responds, "Well, keep trying."